Hello everyone and welcome to Thermo Fisher Scientific webinar series on Sanger Sequencing and Fragment Analysis Made Easy, corresponding with the launch of Applied Biosystems' new Seq Studio Genetic Analyzer. Today we present the fourth topic in the series, Genome Editing Workflow, facilitated by the Thermo Fisher Scientific Portfolio Solution, presented by Dr. Steve Jackson, a Product Application Specialist for Thermo Fisher Scientific. I'm Susie Valdez, and I'll be your moderator for today's educational webinar, presented by LabRoots and sponsored by Thermo Fisher Scientific. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type your questions into the box that appear on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, notice you're viewing this presentation in a slide window. To enlarge that window, just click on the screen icon on the lower right. If you have any trouble seeing or hearing this presentation, click on the support button at the top right of your presentation window, or use that Q&A button and let us know you're having a problem. I'd now like to introduce today's speaker, Dr. Steve Jackson. Dr. Jackson has over 10 years of experience in genetic analysis technology. As a senior member of the genetic analysis R&D team at Thermo Fisher Scientific, he helps develop solutions for the research and healthcare markets, leveraging varied technologies such as capillary electrophoresis, digital PCR, next-generation sequencing, and microarray analysis. Please join us now in welcoming Dr. Jackson. I will now turn the presentation over to him. Welcome, Dr. Jackson.
Thank you, Susie, and thank you everybody for joining today. I'm going to tell you a little bit about how Thermo Fisher Scientific can help you with your workflows that are doing genome editing problems. As most of you know, the genome editing technologies are poised to really revolutionize the way that we understand biology. It will allow us to do many different things, including the things shown here on this slide, anything from making models of human disease in mice and other animals to making disease-resistant plants, even gene therapy research, for example. These technologies, which include CRISPR, Talon, and zinc fingers, can help study gene functions, make gene mutations, tag genes, for other purposes, stably integrating new sequences. Really, almost anything that is biologic can be modified using these genome editing applications. What I'm going to focus on today is mostly the CRISPR-Cas9 system. This is originally a system that was identified in bacteria as a primitive way for them to fight off viral infections. The way that we have made use of the CRISPR-Cas9 system is that the Cas9 nuclease is guided to a particular target by a guide RNA. Once the nuclease and the guide RNA complex finds the target sequence, a cut is made in the genomic DNA locus that has been targeted, and then that genomic DNA, that cut genomic DNA, is then repaired either in a non-homologous fashion, making sloppy changes, or if it's provided with a template, you can make insertions of particular point mutations or GFP sequences, for example, or basically anything at all. So this system is very simple to use, and as I mentioned here on the slide, it makes it possible to make single nucleotide changes, precise deletions and insertions, imprecise or sloppy deletions if you want to knock out the function of a gene. You make deletions at multiple loci. Just basically anything that you want to do, you pretty much can do using these genome editing technologies. Now, Thermo Fisher Scientific, as you know, is a very big company with a very diverse portfolio, and we have been developing these technologies over the course of our last 10 years or so to make things easy for the user to do these type of experiments. Anything from designing the guide RNA that will help you target the right locus in your genome, through the cell transfection, the screening procedures needing to identify the cells that have the change needed, and finally ultimately the phenotypic analyses at the very end, which allow you to study the function of the type of mutation that you've made here. And again, we have anything from genetic analysis tools through mass spectroscopy, flow cytometry types of workflows, and anything needed to characterize the edit that you made in your cells. So to kind of show you how this is going to work, we've done some proof of concept studies. We've made use of the pre-designed human GCD primer database. We have basically designed primers against for CRISPR and TALEN against all the human genes, so you can make sloppy knockout mutations and therefore make loss of function genes. We basically chose some of these guide RNAs from several different genes from this pre-designed knockout collection. We introduced these into 293 cells or A549 cells by transfection, along with the guide with the Cas9 nuclease needed to make the edits. And then we prepared extracts from these primary transformant pools. These pools contain mixed populations of edited and non-edited cells, and we mostly want to try to analyze how efficient was the edit that we designed. How many cells have the edit that we wanted to make? Because the efficiency is key for determining how many cells in the secondary screen you want to analyze downstream. So you want to know how efficient was the edit you made in the first place, and we have different tools available for that. I also want to point out that we also have tools available if you don't want to make use of our pre-designed libraries. We also have the ability to make custom tools or custom guide RNAs for whatever purpose that you want to have for your research. So the first method for determining the efficiency is very simple. It's called genomic cleavage detection by agarose gel electrophoresis. And the way that it works is that you first start off by transfecting your cells. You then prepare a lysate of these cells by just lysing them basically in a PCR-friendly buffer. You then amplify using primers, denature using you basically heat up the mixture and let it re-anneal. When that occurs, you get mismatches of unedited versus edited or edited versus different edits amplicons. These loop-out mismatches are now substrates for a single-stranded nuclease here. And so you add the single-stranded nuclease to this mixture, and where there's a loop-out, where there's a mismatch indicating where there's an edit, then you break a larger amplicon into smaller amplicons. These are then readily observed on an agarose gel. And so what I'm showing here now is the output of one of these experiments where we have the parental band shown. Parental band shown here, shown here. This is the intact amplicon, the full length size that is indicating that there has not been a cleavage event, there has not been an edit, or there has been no nuclease added. But you can see also is that there are smaller cleaved bands which indicate that the sizes of the fragments that are produced as a result of this mismatch that happens here. And by summing up by dosimetry the amount that is in the cleavage products with the amount that's in the uncleaved products, you can get an idea of how efficient the edit was. And in this example here, we have anywhere from 10.8% to 21.5% edit. In a different experiment here, we had a better efficiency, about around 45, 46% efficiency that comes up. So the agarose gels are pretty standard equipment found in laboratories, and this is pretty easy to do these types of experiments. The problem is that with agarose gels, the absolute sizes and the absolute cleavage percentages are not easy to determine because you're relying on dosimetry and imprecise ways of measuring what's there. So another way of doing this is by using capillary electrophoresis. Basically, the experiment works the same way. You transfect using your guide RNA and nuclease. You allow the cells to recover. You prepare a lysate, as we did before. You then PCR amplify, but in this case, you use a primer that has a fluorescent tag at one end. So now your amplicons are going to be labeled at one end. You go through the denaturation, renaturation as before. You do your cleavage using the detection enzyme provided with the genomic cleavage detection kit. And now instead of running an agarose gel, you analyze these by capillary electrophoresis. And the results will look something like this. In this case, we've modified the HPRT locus. We have amplicons that will query this locus here. In this case, we're showing the HPRT amplicon that has no guide RNA added but has had the detection enzyme. And what we get is a single strong peak at about 470 nucleotides that tells you the size of the amplicon that we designed. Now, in the case of no guide RNA, there's no editing event, and so, of course, there's no change in the size or the position of this particular 470 nucleotides peak, as shown here. However, when we take the same cells and now we've added the guide RNA, we get a family of different size peaks after enzyme cleavage. So we have the intact parental 470 nucleotide band. We have a family of bands that are in the range of 229 to 251 nucleotides. And we also have a smaller size here of 460 nucleotides. Now, because this 460 nucleotides appears in the no enzyme capillary, no enzyme lane, what that's saying is that we have deletions of around 10 nucleotides that are pretty abundant. And these are now the uncleaved but different size bands because there's been some of the HPRT locus deleted. In the presence of the enzyme, we can see that the smaller sized bands are indicating that we have a good amount of cleavage, a good amount of edit that has happened here, and it's producing fragments that are around 229 to 251 nucleotides of around where the mismatch has occurred. Now, using our software, it's pretty easy then to calculate the fraction that's present in these cleaved bands versus the total amount that's present in the cleaved and uncleaved. And in this case, we can calculate the efficiency of the edits at this locus as being about 68%. So one thing I want to mention as well is that we just recently updated our software to do this. Classically, we have used a software called GeneMapper, which is a great piece of software, but it's a little bit unfriendly to use. We just released something called Peak Scanner, which is a little bit cleaner interface, but it does the exact same thing that GeneMapper does. And here what we're showing is a different experiment here where we have on the top is the cleaved guide RNA directed edit, and on the bottom is the same lysate in the absence of enzyme. And what we see here is the peak for the intact parental amplicon, and then the family of peaks that are indicating that there has been a genomic edit. And again, what you can do is you can calculate the heights or the areas under the curves to calculate the exact precise genome editing efficiency obtained in that experiment. So the next way that I'm going to tell you about makes use of Sanger sequencing. So in this case, if you take the amplicon and try to sequence the amplicon, what you will get is that you'll find a region that is a pure sequence, meaning this is the part of the amplicon that has been unedited. And at the red arrow that I'm showing here, and downstream of that, you have sequence that is now very poorly resolved. You have multiple peaks that indicate that this downstream of this arrow, there is a mixed population of sequences that are here. So by Sanger sequencing the primary pool, what you can see is that you had a successful edit, but it's difficult to see exactly what is present in that edited pool. You can't confirm the sequence. You can't really tell what the frequency of the edit, the efficiency of that edit was going to be. So there are a couple ways around that. The first way is to basically subclone these into plasmids, and the way this works is that you take your primary pool of cells, okay. After the edit, you would then establish your primary culture, and now you amplify, just as I've told you before, the amplicons surrounding the guide RNA. Now, some of these will have an edit, and some of these will not. You take this mixed pool of an amplicon, as shown here, and subclone it into a topo-containing plasmid. Okay, so now you're basically subcloning the amplicons, establish individual bacterial colonies, isolate plasmid DNA from them, and sequence the bacterial DNA, the plasmid DNA, each of which is going to contain a fraction of the edit that you have in your original primary pool of edited cells. So you sequence these individual subclones now, and then you can then identify the types of edits that were made, and the frequency at which those edits were made by counting the number of counting edits in individual colonies. So here we picked 96 different bacterial colonies, isolated plasmid DNA, and sequenced the HPRT locus here. What I'm showing is that each one of these lines indicates the plasmid sequence, the HPRT locus in that plasmid, from different bacterial colonies. And you can see that there are the large changes that are made, or in some cases small changes made, deletions, insertions, lots of different types of molecules get made when you do these kind of sloppy edits here. On the top is showing the guide RNA sequence, and then the wild type HPRT locus is shown here in blue with the red box around it. The orange or the yellow with red type are showing the sequences that are unedited, that are unmodified in the whole population, and you can see that then that a very large variety of different types of edits were made. So by counting the number of colonies that have an edit and comparing to the fraction of the 96 that contain an edit, now you can get a frequency of the editing efficiency in that particular pool. The other thing that doing this plasmid sequencing tells you are the types of edits that you can expect to see in your edited eukaryotic cells. Now, another way of doing this, which doesn't involve plasmid sequencing, it makes use of software called TIDE. This is called Tracking of Indels by Decomposition, and it was developed by investigators at the Netherlands Cancer Institute in, I think it's in Amsterdam. In any event, what this software will do is it will take a control sequencing trace and a mixed sequencing trace, as shown down here, and it will calculate the frequency at which these mixed pools can be explained by a -1, a -2, a -3, etc. deletion around the guide RNA sequence that you supply to them. So when we do that experiment using the HPRT cells I described for you, what you can see here is that on the forward strand here we have a large fraction here which was shown to have a +1 mutation. A fraction that has a -1, a -2, -3, etc., all the way to -10. And what the TIDE software will do is it will calculate the fraction that are different from the 0. Okay, so the 0 is shown here in the light pink here. These are the fraction of the traces that can be explained by having no edits, and it returns to you a %. It's difficult to see here, but the % here was calculated to be about 80% editing efficiency. We get basically the same profile on the reverse strand here and about the same frequency. So what this tells you is that we can expect that a majority of the edits in this experiment contain a +1 insertion, but that will also be a certain, about 4% will have a -1, about 5% or something like that will have -2, etc. So around 80% of a total of around 80% of the altered sequencing traces are present in this HPRT edited sequencing reaction here. Now we can do the same thing with using a different guide RNA of a different locus. In this case, we chose rel A, and in this case, we got a fraction that was not edited, shown again in pink, and again we have fractions that were either -1, -2, -3, or +1, +2, +3 going the other direction. Got the same results in the forward and reverse strand. Overall, the editing efficiency for rel A was around 20%, so a little bit lower than with the HPRT. And using other techniques that I'll describe for you in a little bit later, I knew that this was a little bit lower. This 20% was lower than what expected. And one thing to keep in mind is that in the TIDE software, you can set the window at which you look for insertions and deletions. Basically, the edit in which you try to deconvolute the sequences that are present. It's set by default at -10. If I expand the window for these rel A sequencing traces, I get a better representation of what the actual editing frequency was. So on the top here, I'm showing the rel A sequencing a TIDE output with the default window, and again, the editing efficiency is around 25% in this direction. But if I expand the window so that I'm now looking at everything from -25 to +25, what I get is what I see now is that there are a larger fraction of sequencing traces now which have deletions of greater than a -10, so up to -15, 20, etc. And if we take into account these larger deletions now, we get a much greater editing efficiency, in this case around 63%, which is more in line with what I've got using other techniques using these lysates. So changing the indel size, the size range parameter in the TIDE software can provide a more accurate determination of the editing efficiency. One final special case I want to point out is if you are constructing a SNP mutation. You're basically expecting there to be only a single mutation or a single change in your edited cells. I'm indicating that here with the red arrow. So if you're expecting a SNP, we have software called Minor Variant Finder, which has been designed to look at the presence of minor alleles present in mixed sequencing traces such as we see here. These screenshots are from Minor Variant Finder. And what we're looking at is at the very bottom here is a control sequencing trace in the forward direction and in the reverse direction. A sequencing traces from a test sample, again in the forward and reverse direction as shown here. And then the output of Minor Variant Finder, which will clean up the background in the forward and reverse direction and look for variants that are present at the same position and in the forward and reverse direction. In this case, we have a G to A mutation, which is in the corresponding C to T position in the reverse strands. For alleles or for variants that have this matched, the Minor Variant Finder software will look for the area under the curve and calculate the frequency at which this allele is present. In this case, this G to A change was present about 12.9 in the forward direction, 10.8 in the reverse direction, so overall about 11% frequency. So what Minor Variant Finder can do for your genome editing experiments is tell you if you have a mixed population, what fraction of those have the edit or have a SNP of interest to you in that particular amplicon. So the last thing I'll talk about is using digital PCR. This is another way you can quantify the efficiency at which the edit is made by judicious placement of TACMAN probes. What I'm showing you here is that we have a particular locus. We have in black a forward and reverse PCR primers here and here. We also have designed a control probe that's labeled with VIC. It's a fluorescent dye, just indicated by red here. And this control probe was placed in a region of the amplicon that is not likely to be edited, not likely to be affected by the Cas9 nuclease and the repair mechanism of the cells that you want to be edited. We design a second probe here using FAM that is over the edited site. Okay? And so what will happen is that in the presence of unedited amplicons where there has not been an edit, both probes will bind and you'll get both a VIC signal and a FAM signal. However, where there has been a genome edit, there's been an editing event that has taken place, the FAM-labeled edit probe will not be able to bind, but you will get a signal from the controlled VIC-labeled probe. So we expect that any kind of the reactions, any kind of the PCR wells that have no cleavage, they will be positive for both FAM and VIC. Where there has been an edit, it will be positive for VIC but not FAM, and there should be no wells that have FAM only because that would mean that the control probe was lost. So as you might know, digital PCR basically will divide a sequencing reaction, or I'm sorry, a PCR reaction into around 20,000 individual PCR wells. We do the PCR and simply count the number of wells that have a VIC probe, a FAM probe, or both. And what I'm showing here are 4 different lysates, 4 different PCR reactions here. And what we get is that clouds of wells, indicating that clouds of positive reactions, indicating the status of the wells that were counted. So in this case here, we have this cloud of cells that were positive for VIC only, this cloud of cells that were positive for both VIC and FAM, and we have this cloud of wells which is unamplified. And by calculating the fraction that is positive for VIC only, as opposed to VIC plus FAM, we can get an idea about what the editing efficiency was. And then we did this basically for 4 different loci and calculated the editing efficiency by digital PCR. So we have presented to you lots of different ways you can quantify the efficiency of genome editing. And what I'm doing on this slide here is just showing you a comparison of the different techniques for the same set of lysates from the same edited cells. So what we have here is in the dark blue bars, we have the frequency calculated by genome cleavage assay on agarose gels. In the orange, the genome cleavage assay on our legacy capillary electrophoresis instruments. In gray, we have the genomic cleavage assay as measured by our brand new Seq Studio instrument. In yellow, we have the editing efficiency as calculated by Tide. And finally, we have the editing efficiency as calculated by digital PCR. And what you can see is that for the most part, the frequencies are fairly agreeable. They are pretty much the same. And there are a couple of exceptions. For example, this GCD agarose is a little bit higher than everything else, and that could have to do with the dosimetry and the other types of methods for measuring the density of the bands in that particular case. In this case, the CDK4 lysate had a little bit lower digital PCR frequency. That could have been because of suboptimal placement of some of the probes, and maybe a little bit of optimization of the CDK4 assay might have done a better job. And in this case, the CHUK4 Tide determination was a little bit lower than everything else, and I know that's because the quality of the CHUK sequencing reactions were not as good as some of the other loci. But overall, the way the different methods calculate efficiency agree fairly well. Now, there are strengths and weaknesses to each one of these different types of techniques, and there isn't really one right answer for determining what the efficiency is going to be. Now, whether you have a knockout, random, sloppy mutation that I just talked about, SNPs, defined deletions, insertions, or translocations, these can all be used to measure all these techniques can be used to measure the efficiencies in these different types of edits, but they do so with differing strengths. Okay, for example, a single TACMAN genotyping assay will be very good at determining whether or not you have a SNP edit made. But for determining what the nature of your knockout mutation is, it's not going to be very good. It can be done, but it won't be very easy or very quantitative. On the other hand, the GeneArt cleavage assay is going to be great for determining your efficiency at producing random knockout mutations, but it's not going to be very good at determining whether or not have a specific SNP present, because you're going to be operating in the background of all these other cleavage events that are present. So really, the method chosen for determining the efficiency of your edit really kind of depends on the type of edits you're making, and not shown on the slide are other things like workflow and costs, amount of sequencing, sequence information you get out of these experiments, etc. So there are a lot of different ways that you can analyze your editing efficiency. That are basically, up to what your question is of your laboratory. But what I'm trying to say is that Thermo Fisher has all the tools necessary for analyzing what those events might be. So to conclude, I have shown you that capillary electrophoresis methods can be integral parts of your genome editing workflow. For determining the edits in your primary cultures, the GeneArt heteroduplex cleavage assay followed by fragment analysis on capillary electrophoresis can determine the efficiency of your edits, but also analysis of Sanger sequencing traces using Tide software or by subcloning those amplicons into plasmids followed by Sanger sequencing will also help you determine how efficient and what is the nature of the types of edits you have in your primary pool of cells. In addition, capillary electrophoresis, in particular Sanger sequencing on CE, is useful for validating the edits in secondary cultures. What I didn't really go into is the next step in this is that if you want to isolate individual homogeneous clones of eukaryotic cells, you sequence or you isolate single cells, and you can Sanger sequence those cells or use minor variant finder to determine whether or not your secondary pools of what you hope are heterogeneous cells contain the edit that you want. Lastly, I wanted to point out that Thermo Fisher Scientific not only has the tools necessary for analyzing the efficiency of genome editing, but we also have the cell culture reagents, the ability to help you design guide RNAs and synthesize guide RNAs, and finally doing the phenotypic analysis from mass spectroscopy to transcriptome analysis on microarrays, etc. So Thermo Fisher Scientific has all the tools necessary for a successful genome editing experiment. Finally, I just want to thank Namrita Ravender on the synthetic biology group for her help in generating the lysates for these experiments, and Kamini Varma and the other members of our content engine team for their guidance on some of these experiments. Also, I thank you very much for your time, and I'd be happy to take questions right now.
Thank you, Dr. Jackson, for that informative presentation. We will now start the live Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on that green Q&A button at the lower left of your presentation window, type in your questions that appear on the screen, and click the send button. We'll answer as many questions as we have time for today. Okay, let's take a look at our audience questions already coming in. Dr. Jackson, here's our first question. I noticed you did not mention next generation sequencing. Is that a technique that can be used for screening?
Can you hear me? There we go. Yes, so next generation sequencing can be very valuable for screening and for subsequently examining the effect of your mutations downstream of that. The issue with next gen sequencing is that the workflow is often kind of complicated, and the data analysis is also complicated, and it can tend to be kind of expensive as well. So again, it depends on the nature of your laboratory and the type of questions you want to ask. Next-gen sequencing can be used to quantify the types of edits that you have made and the types of sequences that you have present. But again, going through the workflow and the expense sometimes is not the most efficient way to go about it. But certainly, next-generation sequencing is a valuable tool that can be used for genome editing workflows.
Thank you, Dr. Jackson. Our next question, what are my options if the genome editing efficiency is low?
So probably the easiest thing to do would be to just redesign your guide RNA and start again. We have tools that will allow you to come up with multiple types of, or multiple sequences for your guide RNAs, so that you have different options that you can choose. You can also try changing your transfection reagents, in other words, using lipofection instead of electroporation, for example, or maybe one of our lentivirus vectors, exclusively designed for genome editing experiments. That might also help change your introduction of the guide RNA into cells. Probably for the best thing, if you continually have problems, you would talk to our synthetic biology group and our experts that are in genome editing for guidance in helping you decide what the best way is to approach your problem.
Thank you, and I want to thank our live audience today. We have some great questions coming in. Our next question, what are some of the selection criteria to choose appropriate technology for primary and secondary screening?
So I touched on some of that during the webinar. I think some of the things that are useful would be to choose the method that is most appropriate for the type of edit you want to detect. In other words, the way that you detect a random sloppy deletion would be different from detecting a SNP. And so the types of technologies used for detecting that, discriminating between those types of changes, be useful for that. But the other important things, are more practical things like, the workflow, the amount of time it takes to generate a result, the cost of the experiment, how much you have to spend to get your answer, both in terms of labor and in materials, the amount of sequence information you want to get out of it. Some of the methods do not actually give you the exact sequence, whereas others like Sanger sequencing or next-gen sequencing do give you an indication of the exact sequence that's present in your cells. So really there are many different variables to take into account for this, and there isn't one right answer for what's best for your laboratory and your experimental system. Again, if you would like assistance, you can contact our technical support group for suggestions on the best way to proceed with your experimental paradigm.
Thank you. And we have time for a couple more questions. Our next one, considering efficiency is important, what are some of the tools available to analyze efficiency?
So again, one of the things that's most important, one of the biggest things you want to determine in your genome editing experiment is how efficient was the edits that you wanted to introduce in your cells made. This talk, went through a lot of different types of methods that are used for that. There's the genomic cleavage detection assay. There is digital PCR. There are Sanger sequencing methods. There's next generation sequencing methods. And in some cases, TACMan might be useful as well. So again, I want to emphasize there isn't one right answer. There are a lot of different ways that you can get at the efficiency calculations. But of course, given how important efficiency calculations are in dictating, how you go about the next steps of your experiment, it really is important to determine what your efficiency is before proceeding.
Thank you, Dr. Jackson. And we have time for one more question because I want to also provide you an opportunity for final comments today for our audience. Our final question, I have heard about the Seq Studio. Could you share some details about this instrument and the applications you can run?
Yeah, so the Seq Studio instrument was just released this year. It is our new all-in-one, easy-to-use capillary electrophoresis instrument. It is designed to be much easier to run and maintain than our previous instruments that we have, and meant to be sort of used at the benchtop, of individual investigators. Some of the applications that can be run besides this genome editing techniques of Sanger sequencing and fragment analysis, there's cell line authentication that can be used. There's clone checking of your plasmids. You just want to sequence whether or not your plasmid construction is correct. There are methods for looking at inherited diseases by fragment analysis. So the whole suite of things you can do on our legacy instruments can also be done on Seq Studio, with the advantage that it's easier to use and in some cases faster. So we're really excited about Seq Studio because we think that it provides a way to democratize capillary electrophoresis applications to our users. And we, again, made it very easy to use and maintain.
Thank you, Dr. Jackson. And I want to thank our audience today. I want to remind our audience members that if they have any additional questions, Dr. Jackson will be happy to email you responses. Dr. Jackson, would you like to provide the audience with some closing comments?
Yeah, again, I thank you for your time in attending this, and we look forward to working with you in your editing experiments, or basically any kind of experiments you're doing. We believe that Thermo Fisher has the expertise necessary to help investigators with basically any kind of experiment they are doing. And if you would like help in your genome editing workflows, please contact us, and we'll provide whatever assistance that we can for you. So again, thank you for your interest and time, and we look forward to hearing from you.
And thank you, Dr. Jackson, and thank you for your important research. I'd also like to thank LabRoots and Thermo Fisher Scientific, our sponsor, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Bye-bye.